Okay, it's 5:01. We might have some folks joining in a little bit, but let's go ahead and get started. Really, just first of all, thank you so much for joining us today for our July Town Hall. We're very excited to have you here with us and to have the chance to share some incredible stories and some progress with you as well. And I'm so grateful to see so many folks joining us today from really, it seems like all over the world, and to see so many different members of different areas of our community come together. I see we've got some EB families on the line, some supporters as well, thank you, and some researchers and folks from the industry and medical communities as well. So just big thanks to everyone for joining. And of course, a very, very special welcome to our amazing speakers, Thomas Murray and Jared DiPalma and Dr. Jose Ayuso, who you'll get to meet very shortly. Just thank you so much for taking the time to speak with us and to share your stories and your experiences. I know we're all really looking forward to hearing from you. Just to quickly go over the format for today, I'll kick things off from the EBRP side of things with a quick but exciting update, and then you'll hear from the DiPalma family about navigating life with EB Simplex and finding your voice to advocate for yourself and your community. And lastly, we'll hear from Dr. Jose Ayuso on his work to combine engineering, biology, and medicine to accelerate the development of new treatments for EB Simplex. Then we'll do our group Q&A session, and finally we'll wrap up with some upcoming ways for everyone on this call to get involved with our community and with our mission. To provide just a quick overview of the format today, we do ask that everyone please stay on mute to respect our amazing speakers. And a quick note that we'll save questions for our Q&A session at the end, as I mentioned. And we're going to prioritize those questions that were pre-submitted when you all registered. So thank you if you submitted a question. But we're going to do our best to answer live questions too. And anything that we can't get to today, we will follow up with you personally after this town hall. So please feel free to send any questions as they arise in the chat, and we'll get to as many as possible in our Q&A. And now, before I pass it over to our wonderful speakers, I just wanted to start with a bit of context on EBRP, really why we're here today and where we're going. So for those of you who might be newer to us, EB Research Partnership, or EBRP, was founded in 2010 with one big, bold, audacious goal, and that is to treat and cure EB as quickly as possible, and in that process, really, to pioneer a path for the 10,000 other rare diseases that are out there, 95% of which currently have no approved treatments. We're the largest global nonprofit funding research solely dedicated to EB, and since our inception, we've raised over $80 million towards this goal, with which we've been able to fund more than 180 EB research projects, leading to just a complete transformation of the EB landscape, including 25 times the amount of clinical trials in the space and the FDA approval of 3 EB treatments. And of course, these are really exciting milestones, but our team is more laser focused than ever on delivering more impactful treatments and eventually cures to our community. And we know that to achieve this, we really need to continue to fund and to accelerate the most innovative EB research from all around the world, which is why we're so excited to announce that our first funding round of 2026 is complete, and we've just approved 7 really groundbreaking research projects. This work spans 5 institutions, totaling a little over $2.45 million, and every single project, whether it be their technique or their target or their technology that they're advancing, every one of those projects has the ability to impact rare diseases beyond EB. And additionally, almost a third of these approved projects were identified by their researchers as by medical definition definitive or curative, which is just really exciting and something that we didn't see even just a small handful of years ago. And as you can see here, these projects span all EB subtypes, and they include a variety of unique approaches, including gene and stem cell therapies, drug repurposing work, biomarker development, and novel disease models. Funding this research will accelerate science that truly will change the world, but it also drives tangible progress for people living with EB today, including those living with EB Simplex, who of course still have 0 approved treatments. Which brings me back to today's topic of life with EB Simplex and progress in the space. So to talk about this important subject, the current gaps in EB Simplex care, and of course the work underway to close those gaps, we've got some incredible speakers here with us. Firstly, we have the DiPalma family. Thomas DiPalma is a young boy from Scarsdale, New York, living with EB Simplex, whose family has become a powerful voice for the EB community. Alongside his parents, Marie and Jared, Thomas has used his experience to help raise awareness and understanding through multiple initiatives. The DiPalma family rang the opening bell at the New York Stock Exchange with EB Research Partnership, which was huge and brought national visibility to the fight against EB. And Thomas has taken that same courage into his own community, presenting on EB to his classmates at school. Together, the DiPalma family continues to speak up, to show up, and to inspire others to help find a cure. With us today, we also have Dr. Jose Ayuso. Dr. Ayuso is an assistant professor of dermatology research laboratories at the University of Wisconsin, as well as an affiliated member of the Department of Biomedical Engineering. His lab's research focuses on studying human disease using advanced in vitro platforms that mimic the in vivo architecture and tissue microphysiology. Dr. Ayuso and his team use these novel technologies to study multiple diseases, including cancer, genetic diseases, and human pathogen interactions. Thank you all so much for your advocacy, your work, and your dedication to this community, and of course, for taking the time to join us today. We're so honored to have you with us. So, without further ado, let's hear from Thomas, Marie, and Jared. DiPalma family, thank you so much again for joining us. Maybe just to start, would you mind introducing yourselves and maybe giving us a bit of a background on your journey with EB Simplex?
Sure. Hi, everyone. Thank you so much for joining us. We're so excited to be able to be here with all of you today, especially on a topic so near and dear to our hearts. So I'm joined, as Allison mentioned today, by my husband, Jared, and our son, Thomas, who will be 13 in just 2 weeks. And so our journey with EB Simplex actually started almost 12 years ago. So Thomas was just about to turn 1 when he was just starting to learn how to walk, and we put shoes on him for the first time, and uh, came up with blisters. And we sort of were like, oh, maybe they were too tight, they were too small. Kept trying different kinds, and no matter what we did, he had these horrific blisters that were forming on his feet. And so we just assumed he had sensitive skin. Went and saw actually 2 dermatologists before anyone even remotely uttered the words EB. And it wasn't until that first appointment where, the doctor admitted she had actually never seen this before. She had only learned about it, had heard about it. And so she was just guessing that that's in fact what this was. And so we kind of left that appointment with more questions than answers. And with some, I would say, some pretty stark statements of, he may never play sports, he may never walk, this is a disease we don't know a lot about, it's very rare. And so we're not quite sure what you're in for, but here's a referral to someone who may have some more answers. And we went straight to genetic testing. That was sort of our first stop. And we were told very clearly that we may not get all the answers, that there were still subtypes of EB that they were discovering, still probably are discovering. And so we may get some inconclusive results. We actually, I think, feel like we were very lucky that we got results that confirmed he did have EB simplex. So at least we had an answer. And then began the journey of sort of what that meant for Thomas and our family. And it's taken on different phases as he's, gotten bigger, as we've learned more, as we've met with other physicians. But we're very lucky to have a doctor here in Westchester who has worked with EB before and has been sort of on our front line in treating Thomas, and not just treating sort of blisters or infections that come up, or but really treating him as a person. And I mean, I think when you go to Dr. Tolugan, he wants to know what you're up to, what you're doing, what camps are you doing, what sports are you managing to play, sort of how is he feeling with all of this, which has been a very important part of this. And I would say we let Thomas guide this journey. I think, I don't know, Daddy and I have not told you things that you can or cannot do. No, we sort of let him guide. So if it's something like a sport that he's playing or an activity that he's doing, if he can hang in there and sort of push through, he does. If he says it's too much, we know it's too much because his threshold is for pain and discomfort is pretty high. So for us, it's really about him being a kid as much as he can be. But I think back to our journey on EB, we quickly found EBRP. I would say within 6 months of his diagnosis, through friends of a friend, which is crazy that an organization that was just starting out, we sort of happened upon this way, and we have not looked back. EBRP has been a partner with us since the very beginning, first as resources and giving me the chance to talk to parents whose children also had EB. The first time I'd ever spoken to anyone who was familiar with this outside of doctors who were still learning. And so we've been participating and fundraising and advocating for this incredible organization ever since. And in the past couple of years, really giving Thomas the chance to lend his voice to it as well, which has been really exciting for us as parents.
That's so wonderful to be able to find that community, hopefully pretty quickly as well. And that's so great, Thomas, that you're able to kind of, listen to your body and really know yourself, and pretty cool of your parents to listen to you as well. That's amazing. I know, of course, every EB subtype is so different, but even within subtypes, really every person is so different. So what have you found to be the particular challenges of your EB simplex, and how are you kind of able to overcome those challenges?
I'll take that. That's for you. Mason.
For me, like the main challenges with EB simplex is just like knowing that if I could do something once, like it doesn't mean like it's not gonna come without like pain at one point. And knowing that like I always have to pay attention to like my body and make sure I'm not doing too much all the time, really, with anything I'm doing. And also just like really just paying attention to my feet and just the rest of my body.
And you've done some different things, right? Are you always necessarily wearing cleats when other kids are wearing cleats? Talk a little bit about what you do there.
Oh no. Really, it's just I always just find like any way to keep doing like sports and like baseball. Like let's say when practice everyone's wearing cleats, like I'm fine with just wearing sneakers, just knowing like I could practice while also not making it that like hard on my feet.
Yeah.
I think you've also done a really good job of starting to like treat yourself.
Yeah.
That when he does have blisters or sees things coming, he knows when to switch shoes or when to like treat a blister. So that's been super proactive versus us keeping an eye. So that's actually one thing that's made it easier as you've gotten older.
Yeah. Thomas just spent 2 weeks at Sleepaway Camp, which has been a great experience for everybody. But blisters are a part of that. And the staff at the camp and the nurses there are fully aware and trained and love having Thomas there. But even with nurses who are there looking and treating his blisters, he still insists that I apparently have a nursing degree, which I do not, and that I can tell whether or not something's infected. So I get photos from the nurses saying, Thomas wants to know if these blisters look okay. But I think to Jared's point, him just having that self-awareness as to how his body's doing and performing, and what he may need to pivot on in order to help get him through that day or that practice. And he'll give himself goals if he knows he's got a double header that weekend. Like he may sit out the practice like 2 practices before to give his body rest and give himself a chance to be in peak performance, right?
Yeah.
That self-awareness is so amazing, Thomas. And not only that, but really the confidence, right? And the independence, that you have to be able to advocate for yourself and, in those situations is really impressive. And I guess kind of speaking of advocating for yourself, I know you've shared with us and my team that you presented on your EB in front of your classmates at school. Could you talk to me a little bit about that and what kind of made you decide to share your story in that way?
Yeah, so in fifth grade, we had to do a capstone project, which was just our end of year like big project, and we got to choose anything that like we really wanted to research and learn more about, and really we like wanted to learn about. So at the time, I really didn't know much about EB. Um, so I decided that that's what I was gonna do my project on. And I really like got to understand what each different type of EB like meant, and like how I was pretty lucky that I only got simplex, and also like how rare it is and how like much of a struggle it is for like other kids who have it worse than me just to do like basic things like walking around. Um, and also I chose it because one, I didn't know much about it, and two, I wanted other people to learn about it more, just so like it wasn't a really awkward interaction whenever someone asked me like, why are you sitting out? Why are you not wearing sneakers? Like all that kind of stuff. I just wanted other people like know kind of what I know.
Yeah, that's a great way to advocate for yourself and to spread that awareness, like you said, not just for yourself, but for everyone else out there living with EB. I'm sure so many people learned about it for the first time that day, and I bet they went home and told their parents, who told their friends, and it's like that butterfly effect, right? Every interaction really ladders up and makes a difference. So thank you so much for doing that. That's amazing. Maria and Jared, I'd love to know, the changes that you've observed in the space since Thomas was born.
Yeah, sure. I mean, I think when we first found out, we were the biggest change is like we have hope now because of the velocity of research and updates we get. I feel like on a quarterly basis at this point. And when Thomas was first diagnosed, there was really nothing, and we just felt that this was just something we'd have to deal with for his life. And now there is so much coming out. It's just a matter of when. Will he be able to participate and be fortunate enough to have some of these treatments for him to make his life that much more accessible to do things. So yeah, hope and change has been such a big difference in the past, I would say 5 years.
Yeah, absolutely.
Yeah, oh, that's great to hear. Thank you so much for sharing that. And then I guess while we're still on you guys as parents, is there any advice that you have to maybe other parents for kind of navigating this journey or for raising awareness in your ways and for rallying your communities?
I mean, I would say that, it to start small. I mean, that's sort of how we started with our participation. I mean, first we took what we could from EBRP as far as having a network and having a support system. But it was, the picture in the middle on the bottom there is I did a spin class fundraiser. I mean, I think we raised like $3,000. We weren't raising seven figures, but it was also something I did locally in my community to, again, educate our friends and educate family about what EB was and introduce them to EBRP. So that's something that's incredibly meaningful to do to make you feel like you're doing something. And I think that's the other thing, to Jared's point about there now being this great hope in the beginning. We just wanted to feel like we were doing something because you would go to these doctor's appointments and other than like treating and making sure he was doing okay, there was nothing else to do. So you kind of felt like you wanted to be as active as you could. And that has had a ripple effect, at least in our community and amongst our incredible family and friends, who have supported EB through, various fundraisers in the past. We have two incredibly brave friends who are actually running the New York City Marathon, with EBRP numbers this year. So we are thrilled to support them on that journey as well. So those are just some of the ways that we have gotten involved and gotten our community and our family and friends involved as well.
That's fantastic. Oh, I can't wait to cheer them on in November. And Thomas, what about you? Do you have any advice for maybe other kids who are, in similar age to you and maybe they want to share their story, but they're a little nervous? Do you have any advice for them?
Um, yeah, just like know that like sharing your story, even if like might be a little nerve-wracking, it's always the best choice because it just brings more like awareness to EB and just to like the other people around you that like that you have it. And also like um um
Why would you want to share? What has
oh
happened since you've shared your story?
oh and since I shared my story, like my friends have also been like more thoughtful about it and just been aware about it. So it's um always just like more comforting to be around them when like maybe my feet are more bad or just like when I can't participate sometimes.
Yeah, thank you for sharing. And it sounds like you've got some great friends too, which is amazing to hear. Before we close up this section, to Thomas, Marie, or Jared, is there anyone or anything, sorry, that you'd like the audience to know that maybe we didn't cover, or maybe something they wouldn't expect about, navigating this journey?
I think the simplex community, it's a unique one in the sense that there's every version is a little different. So there's so much trial and error when it comes to navigating and treatment and what type of shoes or what type of band-aids. So I think the more the simplex community could also be a subset of a cohort that shares information. I think that is really comforting
Yeah.
to us along the way.
Absolutely. And I think, Thomas brought up a really great point. Like we consider ourselves to be very blessed that it is EB simplex. There is so much of this disease that we don't see or have to, grapple with for him. And so to the extent that sort of puts us in this unique position of we are an EB family, but there are other families that have a much different scenario than we have. And so I think that simplex community that Jared mentioned is even that much more important. Like it is okay to say that this is hard. It's a different kind of hard than it is for other families, but just having that community helps so much.
Yeah, I think community is such an important part of any rare disease. And of course, not only EB is a part of that, but EB simplex as well, of course. So that's amazing that you've been able to find that. Thank you so much for sharing and, for everything that you all do for this community, for your leadership, for your voices, for your advocacy, Thomas. It was so much fun to have you at the New York Stock Exchange to ring the bell and to be interviewed on TV. That would have made me so nervous, but you handled it like a champ. So just thank you for everything that you do for this community and with EBRP as well. We're so honored to have you a part of things like this. If anyone has any questions for Thomas and his family, you can put them in the chat now and we'll try to address as many questions as possible in our Q&A session. But thank you, DiPalma family, again for sharing.
Thank you.
Dr. Ayuso, I'll stop sharing my screen then and pass things over to you.
Perfect. Thank you so much, Allison. Yes, I'm going to try to share my screen and hopefully I can do it without embarrassing myself. Okay, so can you see my slides?
Yes, perfect.
Perfect. So again, thank you, for the introduction. I want to also say thank you, to Marie, and Jared, for giving us your time, and especially to Thomas, for sharing your story. As someone from Spain, hearing that you are like soccer, that makes me super happy. I totally approve. So without, anything further ado, I'm happy to, I'm very excited to present at this town hall where I'm going to be talking a little bit about microphysiological systems. I know that's a mouthful, but basically it's about to show you how we can combine different disciplines like engineering that seems so far from the hospital. So we can combine engineering with biology and medicine to hopefully accelerate the development of new treatments for EBS. So the idea basically is that in the last decade, we've seen so many advances in biotechnology and biomedical engineering that now we are building here at UW and obviously other institutions, we can build accelerated pipelines, for drug development, where we can screen new therapeutics that are highly personalized at a fraction of the cost and at a fraction of the time. But I know that's like a lot of details. So before I go into those, let's just start with the very basic, taking one step back. What is epidermolysis bullosa, or also known as EB, right? Very often we say it's a group of rare genetic skin disorders characterized by skin fragility. Yeah, that is true. And EB is caused by genetic alterations in genes associated with skin integrity. I'm not going to go into the details. Probably everyone in this audience knows this. But why am I bringing this up? Because now, as Marie and Jared were saying, the devil is in the details. That's where the details are important. Because depending on the type of alteration and the type of gene that is affected, we're going to be looking at very different types of EB that require very different types of treatments, sometimes even personalized treatment. So I'm just going to use an example from dystrophic EB, that normally affects 20% of the EB community, and we've seen some interesting headlines or promising headlines in the news. Why? Okay, dystrophic EB is caused most of the time by a lack of collagen 7. So, long story short, for these patients, providing more collagen 7 has proven an effective treatment. This is phenomenal. These treatments were developed, by the way, in support with EBRP, so that's phenomenal, and led to the approval from the FDA by some treatments like Biubec and Sebaskin. That's phenomenal for these patients. However, as I was saying, not these different types of EB are very different. EB simplex, people living with EB simplex, they have plenty of collagen 7. Their collagen 7 is working no problem. So providing more is not going to do anything. EB simplex very often is driven by these dominant negative genetic alterations that affect keratins. So in order to develop these specific treatments for EBS, we need to understand the genetic alteration driving EB simplex, if that makes sense. So I'm going to go to Biology 101 in 30 seconds, and see, okay, so what is these keratins and what are these dominant negatives? So these are basically keratinocytes. These two guys here, those are your skin cells, right? They protect you from infections, and so on and what's not. So these cells attach to each other thanks to proteins like keratins, like keratin 14 and keratin 5. A filament of 14 normally binds with a filament of 5 and they form these heterodimers because there are two and they are different. That makes sense. This is a name that makes a lot of sense. These heterodimers, they bind to each other forming tetramers, octamers. I know what's the name for the next one, but I'm sure we can look it up in Google. But eventually they are going to form these keratin filaments. And as you can see, it's almost like a rope, with a lot of filaments that individually are weak. But when you put them together, they can withstand heavy forces. So that's why when you apply pressure on your skin, normally can withstand that pressure, that mechanical trauma. So now you probably see where I'm going. Let's see what is happening in cells that have EBS. So EBS, like we said, they have genetic alterations like this KRT14, blah, blah, blah. You can say, Jose, that's a lot of details. I know. So I'm going to go one step at a time. Basically, in EBS, we have this pathogenic alteration that very often leads to one of these keratins, maybe 5, maybe 14, to be truncated. What do I mean? Maybe it's missing this end of the protein. So when you form the dimer, okay, the dimer is working well. It's missing this tail, nothing too bad. When you go into the tetramer, now you can see that things get a little bit more weird because this end maybe works, but you are missing the tails here to dimerize. So now when you apply these forces, again, when the kid is running, playing soccer, hopefully it's soccer, when the kid is playing and they trip, and that mechanical trauma happens, these filaments are not strong enough, and the filaments just fall apart, that's just break. So that's why what makes the skin prone to that blistering or failure. So now, after a decade of research, we have identified, like again, Jerry was talking about this, that probably not two EBS families are the same. There are hundreds, probably, of genetic alterations explaining EBS. That's, for example, what you can see here. This is the keratin 14 gene. Each dot is a family or a patient that was identified. And it's different, as you can see, different locations, there are going to be different types of alterations. And with the colors, we're just giving you an idea of how severe it is. Some of them do nothing in green. Some of them are going to be moderate. Some of them are going to be more severe within EBS. You can also see that these alterations don't happen randomly. This region has no alterations. Why is that? Because these alterations are happening on what is known hotspots. This one here and this one here. Those basically are critical regions of keratin 14. If something is affected, if something is disrupted here, keratin 14 cannot work, cannot hybridize. So when you apply that mechanical trauma, it falls apart, right? So now that we know this, we say, okay, why am I bringing all this up? Because EB simplex, as we can see, is very heterogeneous. It has all this variability. And drug development is extremely expensive, as you probably know. Here you have some numbers, right? That it takes easily 10 years of preclinical work, followed by another 6 or 7 years of clinical work, just to get one component, one drug approved, and we have to test 10,000. Imagine if now each patient living with EBS needs its own and unique drug. It just makes the system so difficult to support. Why is that? Basically because the tools we use in the lab very often are limited. Human skin is very complex. It's a very elegant tissue. And these are some of the tools we use in the labs. These are the well plates. We call them well plates, basically just cells on a plate. How do you even quantify mechanical integrity on a few cells on a well plate? You just cannot do it. So that's why the preclinical phase takes also so long. So here is where those microphysiological systems can be very, very helpful. Basically, it's just a flashy name for small devices like this one. Those are my fingertips wearing gloves just for scale. So they're pretty small. And maybe you can see that they have some little chambers connected by channels. The idea basically is that we can fabricate a device where the internal geometry that we want and seed the cells that we want. So maybe it's cells from the lung to study lung biology. Maybe it's cells from the kidney to study kidney biology. Or today, Maybe we are taking cells from the skin to study skin disorders. The idea being that traditional in vitro models, like we said, they are cheap and fast, but too simple. We cannot even measure mechanical properties. And in vivo models, like mouse, they're great, but they're incredibly slow. You take even a year, sometimes, or more than that, just to generate a model for one EBS patient, if that makes sense. So it's again not sustainable. This is the gap that we think we can fill with microphysiological systems. They're affordable, but can answer complicated questions. How do we fabricate them? I'm going to skip all the engineering because probably no one cares. I want to focus on the medicine, but there is a bunch of engineering and physics involved in them, so we can build that kidney on a chip or even a brain on a chip, like you can see here. But for today, once again, I'm going to focus on the skin on a chip, where again, I know this is a busy slide. I just want to emphasize this point. Microphysiological systems allow us to generate patient-specific skin constructs. It's almost like an avatar of the patient. So we can test many therapeutics that are highly personalized on a very quickly manner, where the idea is that we're going to take a biopsy from the patient, isolate the different cells, build that skin construct where we can monitor the cells, in the different layers of the skin, like your epidermis, your dermis, your blood vessel. That's what you are seeing here in this microscopy scan. So now the question is, okay, I was talking about how to explore mechanical properties, right? Because that's what we need to do. We need to quantify if your treatment is improving that mechanical properties or not. How do you do that? We're going to borrow again another lesson from engineering, where engineers are really good at building things, like buildings, planes, or even cars. So imagine you are building a Formula One car that needs to go at 200 miles per hour. So the first thing you're going to do once you have your prototype, you're going to put it inside of a wind tunnel, like you can see here, where you're just going to blow air at very high speeds to see what parts of the car are withstanding that high speed. If that makes sense, what we're going to do is now just basically build that for cells. In some of these MPS, what we're going to do, now we're going to have your keratinocytes, and we're going to flow air or liquid at a given speed. And because we know the mathematical principles that drive this, we can calculate how much stress those cells are suffering. Before the math gets boring, I just want to show you this. These are basically one of these microphysiological wind tunnels. Maybe you can see some dots moving bottom to the top, right? That's because we're applying that airflow. We are flowing air or liquid. So that's why you see those particles moving. You see these things here? Those are skin cells. These are your keratinocytes. One, here you have another one, this is another one. And basically you see that hopefully at the top you can see the time. You can see that I'm flowing air, very high speed. These keratinocytes don't care. They don't move because they are very strongly attached. That is great. That's what we want to see. So now I want to show you what happens when we have keratinocytes, that harbor these EBS alterations. Whoop. You can see that when we apply the same level of pressure, now you see what's happening to these cells. They are just flying off. They are not strong enough to withstand that pressure. And this is about after 30 seconds, but you can see how after about a minute, most of the cells are gone. So this is what, now we have a technique to quantitatively and in a personalized manner analyze how much treatment you need, how much you need to correct. So hopefully with this, now you are becoming experts on engineering. I'm going to move to the medicine. Like I said, I want to focus on that. The analysis of this is basically based on AI. These days, these are great tools, like machine learning to quantify the number of cells that detach. Again, that's a technical details. I'm going to focus on the medicine. Some of these treatments are gene therapy. What specifically are we doing here? We're doing base editing. What is that? I'm going to explain it very quickly. In UW, now we have a clinical protocol where we can collect samples, from people living with EBS, and we're going to store them in a repository. So the idea is the following. We're going to have the EBS patient maybe send some saliva. Very simple, minimally invasive. We're going to do the sequencing of maybe keratin 14, maybe keratin 5, and we're going to look at the different sequence until we find that genetic variation that is explained in the EBS. And we say, okay, it's here. Now, if it's compatible with the treatments that we're going to explore, we say, okay, we can get some cells and we can quantify the levels of keratins. That's what you have here in green. These are your keratinocytes. This is the wild type with high, bright green signal. And here you can see the green signal from this example, this patient had a mutation, had an alteration that is lowering the levels of keratins. And as you can see, the signal is very faint. So we want this signal to go up, okay? So how do we do this? This is where now I explain how these genetic alterations can be read. So this genetic alteration is affecting maybe keratin 14 at the location 374. Normally you would have a 1 in, that's a G. That's wild type allele. This is what should happen. But in this patient, there is one allele that instead of a 1 in has an adenine. So that's why this is labeled as A. So these patients very often, one of their alleles, one of their genes is working perfectly fine. We want to protect this. But the other allele is the pathogenic one. This is the one that has the genetic variation. In this case, instead of a G, we have this A. How do we fix this? With a base editor. This is a molecule that basically can take this A and transform it back to a G. So now, once that's happening, the pathogenic allele, it's exactly the same as the wild type. So this would be a permanent cure for this type of, alteration. How can you modify only one of those nucleotides, one of those letters? We do that with patient-specific molecular probes, where the idea is that we are going to take a look at your profile, we analyze where the genetic alteration is, and we design a probe that is going to maximize the editing of that specific location. Some of these ones, for example, are very, very compatible, achieving like a 90% efficiency. Some of them are going to be maybe on the 80%. Some of them are going to be on the 35%. So for example, this one is not that compatible. So what do we do for this population that is maybe not that easy to fix in vivo? So the idea for this population is an ex vivo approach where we're going to take the cells out of the patient, we engineer them with base editors, and you transplant them again, using a skin graft. This is exactly the same strategy they are using in Zeva skin for dystrophic EB, that again was approved recently. So this is again for alterations that are poorly suited. For other ones that maybe are better suited, that 80%, 90% that I was talking about, here is where we can explore in vivo correction. This is where things get even more interesting, because now we don't need that grafting anymore. You could just apply the base editor on the skin, or maybe systemically through an IV, and you're going to do the correction in the patient. This is exactly the same treatment that was used for the first time in the US in humans for this little fella that you have here. This is baby KJ that has a disease driven by another dominant mutation. And as far as I know, 1 year later, he's doing pretty well, and the results are very promising. How do you deliver this base editor? Normally, we used to use viruses. That's the kind of the same technology that you see in VQVEC and whatnot, which again, they're great. But now I think we have a better technology. So I'm going to talk a little bit about lipid nanoparticles. These are basically oil spheres that can have inside that base editor, so it's protected, so we can deliver them to your cells. If this maybe sounds a little bit far-fetched or science fiction, this is exactly the same technology that you are using in the COVID vaccine and the flu vaccine. So if at any point you got these shots, that's how this is done in the clinic today. So here we are partnering with the Weizmann Center at Wisconsin because they have the facilities to manufacture these lipid nanoparticles on a pharmaceutical grade. So anything they manufacture is very close to be delivered in the clinic. So I want to show you a little bit of the results that we are getting with this. Our nanoparticles are green. So if the cell is taking the treatment, you are going to see the cell turning green. So here you can see EBS cells before the nanoparticle. Maybe you don't see anything that exciting. Here you have the results after the nanoparticle. You see all these green dots? Those are those keratinocytes that you saw in black and white before. You can see that glowing very bright. So here we're showing that about 50% of those keratinocytes, those skin cells affected by EBS, are taking the drug, which we were very happy, but we thought we could do better. So we run an optimization pipeline where we were tweaking the nanoparticle. I cannot disclose. The legal team told me not to disclose this because that's the way to protect this. So it goes into the clinic. So I have very boring names like UW, LMP14. But this has been optimized, and now we are achieving more than 90% delivery in these cells. So 9 out of 10 cells are being edited, or at least we're delivering the base editor to those cells. So we were very happy, to see these results. I'm also going to talk, like I said, we're exploring different therapeutics. There are some EBS alterations that will not be compatible with base editors. What do we do for those patients? Like we said, we don't want to leave anyone behind. So that's what we consider something a little bit different, something like an antisense oligo. This is called an ASO. What's an ASO? Very, very, very simple. Remember our example for this patient, right? That it has a wild type allele that is working properly in green and a pathogenic allele in red. This is the one that is causing EBS. This is the one that we want to stop, from causing damage. So an antisense oligo is basically a patient-specific DNA probe, this one in green, that is going to bind to the pathogenic allele. So now this guy cannot express, cannot do anything, is going to be labeled for degradation, and is going to do no damage. Because the green probe, the ASO, is different from the wild type, it's going to spare the wild type. So the wild type can still work and produce enough keratin for this patient, to rise the levels of keratin. This at least is the theory, right? I just want to also bring up, that this is not against science fiction. This is approved by the FDA for other disorders that have dominant mutations, very often in the neurological space. Think about like amyloidosis, sclerosis and whatnot. These are just some examples. So we want to bring this technology that is already partially validated from these neurological disorders to EBS. How we design this? Again, this is the concept, right? We're just going to design the ASO, matching the sequence. This is a little bit simplistic. This is not how DNA or RNA looks in vivo. This is how it looks like. It's a very complex 3D structure that is very difficult to predict how we're going to attack if my ASO is going to be able to reach the core that needs to be destroyed. So this is, again, where we take a lesson from engineering and AI. We're going to run, machine learning driven accessibility analysis. It's a mouthful, I know, but basically what we're going to do, we're going to analyze how easy it's to attack the pathogenic allele and the wild type allele, right, on both of them. So this is the accessibility profile. Now you are going to match them. And now we're going to say, okay, let's try to identify, these locations where we can attack the pathogenic variant while protecting the wild type. If I'm explaining this in very simple, it's just because this slide is very complex. This is how it looks like in real life. This is a machine learning driven ASO design for EBS, where the computer is identifying what is the best spot to attack the wild type, to attack the pathogenic allele and protect the wild type at the same time. So these are some of the lead compounds that we identified. And this one in green, we said, okay, this is the one that should be the best one. So let's go for it. So here I want to show you just like a glimpse of some of the results. These again are these patient-derived EBS cells before the treatment. Remember, for this patient, what we were seeing is low levels of keratin 14. That's the green signal. You can see the cells, they're kind of faint. So here you have the results after 5 days, treated again with boring UW ASO number 9, where you can see how these cells are turning brighter. The more green they are, the more keratin they have. So you can see how in 4 days, we're starting to see a rescue, at least partial, of that keratin 14. We still have a long road ahead of, dosing, timing, blah, 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 but that was, very promising. And I'm just going to close with about the last leg of this program that we have, that is about drug repurposing. Genetic therapies, ASOs and base editing, they are great, but they probably still need some years in the oven to be delivered quickly, in patients. So in the meantime, we're exploring drug repurposing. As you can imagine, there are other diseases, or other patients that have to deal with chronic wounds. Think about people living with diabetes. Very often they have chronic ulcers that don't heal properly. So we have FDA-approved compounds for those diseases. We can, for a fraction of the cost, use these microphysiological systems to see if they would be relevant for EBS. So we can have a much faster development pipeline, a lower cost. And again, we can just use this microscopy analysis where we can see these EBS keratinocytes healing this gap, healing this wound in real time when we treat them with these compounds, with these boring names, UW001, that again, I have to protect, we basically use the same machine learning analysis. And I know it's a busy slide. I just want to give you with one glimpse again at what we are doing. This is basically how much of that wounded area is closed at a given time, from 0% to 100%. You can see that after 2 days, when we don't have any treatment, it's 40%. With UW001, we can see an increase up to an 80%. This is almost double the healing speed of normal cells with no treatment. So we are very excited about how we can use these microphysiological systems to really, really speed up this preclinical heavy work so it gets into the clinic faster. For this UW001, we think we're pretty confident. We think we have a strong position. So we think we will be starting maybe late 27, early 2028, a phase 1b clinical trial for people with EBS. Maybe we expand it to EB, other types of EB. ASOS, like I said, we've done the silico design, the delivery. We're starting to see the first evidence of rescue of keratin. Remember, those were those green cells getting greener and greener. So hopefully this is still, we need a few years. And base editors, this would be basically the pie in the sky, in the sense that this would be a curative, permanent, one-off treatment that if it works, and we still need some years, for the preclinical work to demonstrate that it's feasible, we could provide that permanent cure. So with that, basically, I'm going to say that we obviously have other treatment, another leg of this research program where we're exploring treatments for EB-driven cancer. And this is a nice segue on my last slide, where basically I want to say that this is probably a lot of work busy with math, AI, nanoparticles. It's because we have a highly multidisciplinary team. I didn't do most of the work. It's the students, doing the experiments Monday through Friday and Saturday and Sundays, like Seth and Friedrich. And also we have a lot of people in UW, like Dr. Andy South, who's an expert in EB, driving all the clinical translational. We have Chris Saha, who's an expert on genetic sequencing and base editor. Jax, who's helping with the cell manufacturing and LNP. Sandro, who's helping us again with delivering the LNPs to the cells. And we have a massive clinical team, with Dr. Bedrole, Dr. Lisa Arkin, Christine, and Kayla, providing care for patients and helping us to identify patients and whatnot. So this is an example of how a team that maybe not all of them even knew about EBS 2 years ago. I want to say that thanks to the support from our funding sources, including EBRP, now we have so many different people with so many different backgrounds, working together for EB. So I want to believe that, we're making progress for sure. And again, like I said, this is my last slide, so I'm open and I'm very happy to take questions.
Incredible. Thank you so much, Dr. Ayuso, for just the amazing work being done at UW and for taking the time to talk us through it today. It was so informative and very exciting too. And I just have to say, it's so impressive to be able to talk about these very complex topics and programs in ways that are digestible and understandable to the everyday audience. So thank you. I know that I learned a lot, and I'm sure everyone on the call did as well. So thank you again. And before we dive back in and get to your questions for our Q&A session, I just wanted to take a moment to shout out some very important members of our community who are on this call. Their family is also affected by EB Simplex, and they've really rallied around our mission and this research and the University of Wisconsin in just beyond an incredible way. So a big, big thank you to the Oros family. We are so grateful for your support and to have you here with us today. So maybe let's all give the Oros family a big round of applause, and to our speakers as well for amazing presentations. Thank you, guys. Fantastic. I'll get my slides back up here and we'll dive into our Q&A session. Just as a reminder, we're going to start with those questions that were submitted when you folks registered. But if you have any questions at all, just pop it in the chat and we'll certainly get to as many as we can. Dr. Ayuso, we'll stick with you for just a moment. Someone was asking how long before scientists will be able to edit genes to spare future generations from being affected by EB simplex?
Yeah, that's a great question. So I'm going to divide it just super quickly into answers, right? For example, if you're thinking about the next generation, that's something that is even done today in clinics that specialize in IVF, right? Like in vitro fertilization. So if, for example, you are concerned about that, you can go to one of those clinics and they can help you to ensure if you are a person living with EBS, they can help you to be sure that your son or your daughter don't carry necessarily that pathogenic variant. A more interesting question is when are we going to be able to fix that pathogenic variant so that patient doesn't need to obviously live with that? Like I said, it still probably needs a little bit more development in the clinic, but like you saw, in 2025, the first patient was treated with those base editors, for a dominant negative disease. So it's just about bringing that concept now to EBS. So I want to believe that, again, maybe in just a few years, we can start to see some of that implemented. Like we said, the devil is in the details. I don't want to, give like false hope or anything. We need to see those sequences, see how compatible it is. But the technology is starting to appear in the clinic these days. Hopefully that's a decent answer.
Yeah, that's fantastic. And that's so exciting to hear. And I think a lot of that, progress and the fact that we're seeing it in the clinic is thanks to you and people like you at UW. So thank you for that work. For the DiPalma family, someone was asking, really based on your own personal experience, what do you feel are still the biggest needs of the Simplex community, the biggest gaps in care, and how can everyone on this call rally together to help fill those gaps?
Want to take it?
I go back to what I may have mentioned earlier. It's just information.
Mm.
I think because everyone has such a unique situation, it affects Thomas's feet. For others, it could affect other parts of the body. It's sharing information and tips and tricks that we
Yeah.
all use on a daily basis to make living with EB as tolerable as possible.
Yeah, I mean, I think I'm going to let Dr. Asos and his team do the hard work from a medical treatment standpoint, but I think it's the day-to-day stuff. It's the like, hey, I found shoes that seemed to work for him when he was, 2 and learning to walk that were comfortable. I mean, we've run the gamut of whether it's like certain types of clothes that feel, more comfortable on his skin or types of bandages and just a place and a repository on where to share that information and search for that kind of information, I think is still something that would be useful to that community for sure.
Yeah, absolutely. That's definitely a gap to get that, kind of like vetted information and information that's particular to Simplex, like you were mentioning. Absolutely.
Yeah.
And Thomas, a question for you. Someone was asking, and I know you mentioned soccer and baseball, which is awesome. What do you like to do in the summer, and how do you stay active, especially in the outdoors when it's, hot, or you might be prone to more blisters? How do you like to stay active in the summer?
I usually like to play sports, only every once in a while, just so my feet don't get too bad. Because as you said, like with the more heat, it just adds up to just more blisters and easier to get the blisters. So play sports or just like sit outside and just like um kind of do anything that's just easy to do or just something that entertains me for a little.
Yeah.
Yeah, what about, I'm so curious, what did you do at camp? What was your favorite activities there?
Probably sports.
What did you play while you were there?
I did a lot of baseball, so that was pretty fun. Um, but yeah, just like other camp activities like color war. Um,
Oh, cool.
but yeah.
That's awesome. Thank you for sharing. Dr. Ayuso, back to you for a question on eye complications. Somebody had mentioned when they RSVP'd, they feel that they found very little information specifically on the eye involvement in EB simplex. So they were wondering if there was any evidence or guidance or patient resources that you're aware of on the ophthalmological complications of EB simplex.
I see. So that's a great question, right? The eye complications are common in EB in general. I think in EBS, they are less frequent, but true, it can happen. I think it's around a 10% or something like that, which is way lower than other types of EB, like dystrophic and whatnot. But they can happen. So for sure, we need to be aware and acknowledge that they are there. Finding information, because in EBS, they are more rare. I don't think I know a unique resource for EBS specifically eye complications. Normally they are grouped together in this case again. So you would see websites where they talk about, ophthalmological care of the eye in EB, but it's in EB in general. So again, that's where the devil is in the details. So I would say that depending on, probably the ophthalmologist is going to be the best person. And then if you have that genetic information, about the genetic profile, that helps a lot. So we have an idea of, depending on the gene that is affected, that's going to inform a little bit what we could expect. Because for whatever it's worth, there are treatments, for the most common symptoms associated with EB in the eye, like corneal erosion, dry eyes, and even blurry vision. There are things that we can do.
Okay, that's great. Thank you so much for sharing. And to whoever answered or asked that question, I can follow up maybe with some additional resources as well after this. And I think based on our time left, maybe one last question for you, Dr. Ayuso. And I saw you responding to someone in the chat, so thank you for looking at that as well. Are there any updates that you're aware of that you're able to share on any clinical trials in the EB Simplex space that are currently ongoing?
So for EBS, there are a couple of clinical trials. I think it's in Northwestern and Stanford, where they are testing. Basically, it's a topical treatment. It's a modified mTOR inhibitor. I'm not going to go into the details. It's a topical cream that promotes wound healing and tries to protect and stabilize keratin. So it kind of promotes degradation of that keratin that is not working and protects, to some extent at least, that wild type properly working keratin. So that one was started in 2025. I can find the name, obviously. And I think they are also organizing another one that started, or at least it was going to start in early 2026. So I can obviously share those resources. I think, again, I'm going to make a shout out to your website, Allison, EBRP, and also Deborah. They make a wonderful job at keeping lists with clinical trials that are ongoing. I think these two that I mentioned, they are both for EBS and other types of EB in that sense. So they are not uniquely, but the treatment could work and should work for EBS.
Great, thank you so much. And yes, that's true. We do have a page of clinical trial updates. As soon as we're aware of them, we do try to update our website as well. So yes, you can always feel free to check that out, or to email me or anyone on the team personally, and we'll try to help you find that information as well. Okay, so with that, thank you so much again, Thomas, Marie, Jared, Dr. Ayuso, of course, and to all of our attendees as well for your participation in today's session. Just to quickly wrap things up, I wanted to share a few upcoming initiatives that might interest our community on this call today if you're looking to get involved. As you may have seen, if you follow us on social media or you get our newsletters, our landmark event, Venture into Cures, is back and will be taking place on November 11th, right here where I am in New York City at Pier 60, and tickets are now live. We were guided by this year's theme, where stories take flight. The evening will unfold through the voices of the EB community, live music, performances, art, and more. And we're just really excited to gather the whole community together in person, including doctors, researchers, supporters, our biotech and pharma partners, and of course, those living with EB and their families. Speaking of, you are the stars of the night. If you are part of the EB community and you're interested in attending, of course, there's no expectation, to sponsor or purchase a ticket. Your seats would be complimentary, and our team would be honored to welcome you. So if you are, a member of the EB community in that way, please feel free to reach out to info@ebresearch.org, and we'll take care of the rest. Or for anyone else that's interested in attending or sponsoring, you can learn more at the link that we'll drop in the chat. And secondly, you heard me talk a little bit about the cycle's exciting research updates, earlier in the night, but really there's so much more to explore about this innovative and exciting work. So if you would like to dive in and learn more, you can visit the link in the chat, really explore each project a bit more in depth, along with the research teams behind it. And lastly, as it always goes, of course, as one research cycle closes, another one opens. So if you're a member of the research community, definitely stay tuned because our second and final research cycle of 2026 will be opening later this week. So in the meantime, you can visit the link in the chat to learn more about our process, our general guidelines, and to access the application once it is live. But with that, just thank you so much again to everyone for taking the time today to join us, to ask questions, and to learn from our fantastic speakers. We appreciate each and every one of you, and I hope to see you next time. Have a great night. Thank you so much.
Bye, everyone.
Thank you, everyone.
Thank you.